So let's now go back and make sure people understand the structure of a muscle, because I want to talk about different
Okay.
fiber types as well, just to round out some of the physiology. Um, so, in an effort to understand the difference between a fast twitch and a low twitch, fast twitch and a slow twitch muscle fiber, which has a metabolic difference, I'm curious as to what the structural
Hmm.
difference is, and maybe just kind of explaining how myofibrils work and things like that.
Yeah, sure. So let me go back just a little bit to understand whole human function, movement. I won't go as deep into this one though. If you just think about how you actually create movement, it really has three core functions. So number one, you have to have some sort of direction or signal. And this is going to be coming from your nervous system. And so whether this is central or peripheral, whether this is autonomic, whether this is a controlled somatic action response, it doesn't really matter for this conversation. The nerve has to tell it what to do. So if nerves, you get that one. That's it. Don't take anything else, nervous system, right? You get that control. Now that nerve then has to go into a muscle fiber and tell that muscle fiber to contract. Okay, the muscle fiber then is part two. So the cell actually has to contract itself, but that actually doesn't cause movement. Muscles are not attached to bone. That's not how it works. So muscle fibers are surrounded by connective tissue. All those connective tissue are bundled together in like a package. So if you imagine buying a bunch of strips of bacon from the butcher and they would wrap that up and kind of saran wrap together, that's actually kind of what a muscle looks like. So you've got that saran wrap connecting it. So if you pulled on one piece of bacon, you'd notice the whole package moves. And that's sort of the point. You're transferring force from muscle through connective tissue. That connective tissue comes together um, into a tendon, and that tendon then attaches to bone. And so the third part for human movement is actually connective tissue. And so you have to have a signal, you have to have a muscle contract that has to make connective tissue pull on a bone. That actually is what generates human movement. Well, if you look at the front end, we'll leave the neuroscience to other people. You look at the connective tissue, and it's very difficult to understand what's happening there for a number of reasons, but mostly it's not plastic. When we look at muscle, it's tremendously plastic, and what I mean by that is it adapts, it changes very quickly and rapidly in response to a lot of things. Connective tissue doesn't have a blood flow supply, it doesn't have an energetic demand. It's kind of just there. There's more to that story, but we'll just kind of leave it like that. The core of the issue of adaptations, whether they are pro or negative, is going to be in skeletal muscle. And so here's what that actually looks like. A nerve will come down and actually attach and innervate to a whole host of muscle fibers. And so you can imagine skeletal muscle fibers are some of the largest cells in all of biology by diameter. They're tremendous in humans. In fact, what's actually very interesting about humans that makes us special is our muscle fibers are what's called multinucleated. And so you probably remember this term from like med school or something like that. In fact, whenever I talk to biology people about this, that like their head is blown because I forget how lost in exercise science I get, with like us nerds. But it's very uncommon in nature to see cells that have more than one nucleus. And the nucleus is the core of the cell, if you will. It's what holds your DNA. That's what controls and tells you when to replicate proteins, to grow, shrink, die, repair. So the whole control center. So the fact that skeletal muscle has many of them per cell, in fact, it's not a few, it's not two or three, it is thousands per cell. So skeletal muscle can be extraordinarily large. You can actually, I have a video of this somewhere, I can't remember. Actually, there might have been a picture in a men's health thing we did. There's a video somewhere
Okay,
where I
well,
actually pick
we'll
up,
find that and we'll also put that in the show notes.
okay. I can actually pick up a single muscle fiber from a human with tweezers, and you can see it with your naked eye. So we could hold this. In fact, I could do it right now if I had one. I could hold it in front of the camera. You'd be able to see an actual whole muscle cell. They're that large. In fact, they can be very, very long. So they can be several inches in length.
And let's help folks understand what defines a cell, because normally outside of the muscle, we kind of define a cell by a cell membrane, has a single nucleus. I mean, we kind of know what the constitutive elements here. This is defined also by a cell membrane, but it's a sort of a longer looking tube as opposed to a sphere. And
Oh, yeah.
yeah, just sort
Yeah.
of,
Thank
yeah.
you. Yeah. Yeah. Good distinction there. I sort of, again, get lost in exercise science here. So yeah, if you remember like you're back to high school biology and you think of a cell as like a circle or an oval, this is more, it's like that. It's circular, but it's a tube. So it's a very long tube. The way to think about it is like a ponytail. So if you think about a ponytail, you think about it as one thing. It is a ponytail, but it's made up of a whole bunch of long tube individual hairs, and they all wrap together to make a ponytail. That's what a skeletal muscle cell looks like, which is actually quite different than a cardiac cell. Those are more rectangular, if you will. That they're shorter and wider. Skeletal muscle fibers are very long, very narrow, but still circular. They still have a cell membrane. They have a bunch of nuclei. Most of the organelle are the same as any other thing. The contractile units we can get to in a second, but yeah, that's the basic setup of them.
And just give me the typical length of a muscle cell, a skeletal muscle cell.
You can't really give a typical, because depending on what you'll see with skeletal muscle is structure is function. So if you can contrast this to cardiac tissue. So cardiac tissue is actually quite interesting because it is what we call the ultimate slow twitch fiber. And so all of cardiac tissue is slow twitch. And in fact, the slow twitch are even more slow twitch than the skeletal. And they tend to be fairly uniform. So you could give specific numbers on cross-sectional area, a diameter, length on those. Skeletal muscle, if you look at like your sartorius, which is that kind of muscle that goes from that pointy part of the front of your hip down to the inside middle of your knee, theoretically, those fibers could run the whole length.
A single cell.
Correct.
Can run that whole length.
Yeah. Even if it runs half that length, that's extraordinarily long, right? You go back though, and you go to like an ocular muscle, then it's going to be minute. It's going to be extremely small in length. If you go to muscles in your digiti, your fingers, they're going to be very, 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 very short. So it's sort of, there is no like classic range. It could be from millimeters to literally inches in length.
So so presumably the reason that these cells have multiple nuclei is, basically to decentralize the actions of cellular construction, right? So you've got DNA making RNA in the proximity of that nucleus coming out onto the Golgi making protein. And if you had, for example, even a 1 centimeter long cell, which is enormous,
outrageous,
you couldn't simply make all of that work with one nucleus. So the question is, does that mean these nuclei act independently? Like, where's the central command on this? It seems like a remarkable problem.
Okay, boy, remarkable problem, remarkable advantage, right? It's the same.
Yeah. Hard to control, but amazingly adaptive.
Exactly. This is exactly right. So if you want to dive down the entire nucleation question, this gets very, very interesting, because a handful of things. We've actually shown in our lab that a lot of professional athletes have more nuclei per volume. And so this is one of the things that I posit is maybe this is why they can adapt so well, why they can handle the volume that they can handle, is they just simply have more of these nuclei around.
And you believe that is how much genetic and how much adaptation to training?
Boy, I would love to give you an answer there. We have, like anything it's going to be. Okay, so there's going to be a component to both. We actually know of numerous lifestyle factors that are going to influence these things, but the more recent data are showing this. In general, we have thought that nuclei have a couple of things. So one, it's not just nuclei count that matters, which is what we previously thought. The shape matters. And so there's like spheres, there's ovals, they're all kinds, and it looks like the shape determines the function. The location determines the function. And so it looks like there are subtypes of nuclei that surround, for example, the mitochondria. And they're going to be very specific to mitochondrial repair. And then there's other types that are more specific to the periphery that'll do cell wall damage. And then there are some actually that are regulating injury specifically. This is what it looks like right now. And so there are subtypes, and this is very, very recent understanding. And this is probably why some folks recover and respond to injury more than others, is they just simply have more of this subtype. Now, to your question of nature versus nurture, what's challenging about that is the measurement fidelity here is difficult, and the tech is moving quickly, but it's sort of like every couple of years when the microscopes get better, we sort of realize that all the 3 previous years are now invalidated. And so there's just a lot of movement back and forth. And in fact, If you look at this related to cell growth, in other words, hypertrophy, there seems to be tremendous confusion about the role of these things in growth or not. So there used to be a thing that we referred to as a myonuclear domain limitation. So in other words, a cell would only grow, so this would be your fiber, it would only hypertrophy or grow in diameter to the extent at which the nuclei could control it. And so in order to gain more growth, you actually have to get more satellite cells to come in and add nuclei, right? And then when you detrain, that cell goes back down in diameter, but you preserve the mononuclear number. And so then now retraining is easier than it was the first time.
So, I mean, this is unbelievable, but this is the old adage of, muscle memory, quote unquote. It's easier
Bingo.
to regain muscle you once had than to put on muscle you never had. And I've never heard a molecular explanation for that, but if that's a very plausible mechanism, right? Which is you
Now it looks like
retain the
that's not
nuclei.
correct, though.
It looks like that's not correct. Interesting.
So
Okay.
there's it's very it's very back and forth is the way I'll say it. So something is there. The story I just outlined to you, it makes intuitive sense. I got really hot on it for a number of years. And then it was some more challenging data came out and it was well, we don't think so. I'm just gonna have to say
TBD.
This is every week another paper comes out and it's just okay, now we're back on it. Now we're back off it. And now we realize there's subtypes of myonuclei and they're oh shit, okay.